Hello students, today biomolecules continuation. So biomolecule lesson continuation, today we are discuss about peptide bond formation. Peptide bond formation. Amino acid linked with another one amino acid. Covalently bonded. In these types of amino acid is known as peptide bond. That is one amino group, one acid group. First number one amino group, that is NH2, and one acid group, COOH. One amino group, NH2 group, and one acid group, COOH group, linked to form amide linkage. In this bond is known as peptide bond. So peptide bond further classified kuda. So dipeptide, tripeptide, tetra, penta, etc. More than two peptide bond is known as polypeptide bond. Polypeptide bond. Okay. First number one. NH2, CH2, COOH, glycine. So glycine is reactive with the alanine. NH2, CH, CH3, COOH. So glycine is reactive with the alanine. So glycine is reactive with the alanine. Removal of water. So removal of water replace. The remaining product NH2, CH2, C, double bond O, N, H, CH, CH3, COOH. Next go to glycine. Glycine amino group COOH, CH2, NH2 plus alanine acid group this glycine alanine. So glycine amino alanine. So alanine C terminal alanine. This is C terminal. Glycine N terminal. Group. Glycine is a N terminal. Next alanine is a C terminal. Removal of water molecule. COOH, CH2, N, H, next C double bond O, CH, CH3, NH2. It's a dipeptide bond. Between COOH group is reactive with the amino group. This is a peptide. It's a dipeptide bond. Okay. Next go to classification of protein. Okay. Classification of protein. There are two types of classification of protein. First number one is a fiber proteins. In these types of protein becomes a linear molecules fibers. The structure becomes linear. So linear molecules is a fiber. First number one. Second one, in these types of protein, it is insoluble in water. This is insoluble in water. Water insoluble is a fiber protein. Point number two. Third one, disulfide bridge. Disulfide bridge. So disulfide bridge, the sulfur is contained. Bridge. Fourth one, fiber protein is contains intermolecular hydrogen bondings are present. Intermolecular hydrogen bondings are present. Example, fiber protein is example, keratin. Fiber protein is example, keratin. Fiber protein is contains kuda. First point number one, it is a linear. Second one kuda, it is insoluble in water. Third one kuda, disulfide bridge is accompanied. Fourth one, presence of intermolecular hydrogen bonding. Presence of intermolecular hydrogen bonding. Fifth one is examples for the keratin. This is a one type classification fiber. Second one is kuda, globular protein. Just reverse. So fiber protein is contains kuda linear, but globular protein contains overall spherical shape. So, spherical shape. First point number one. Second one, it is soluble in water. It is soluble in water. Third one, it contains a polypeptide bond. It contains polypeptide bond. Fourth one, many of the functional groups are present, especially enzyme catalysis. Especially what? Enzyme catalysis. Fifth one, example, myoglobin. Fifth one, example is a myoglobin. First number one, so overall is a spherical shape. This is a spherical overall. Overall is a spherical shape. Second one, it is soluble in water. Water soluble protein. Third one is a polypeptide. More than 2 peptide bond. More than 2 peptide bond. Polypeptide. Fourth one, many of the functional group can accommodate. Many of the functional group can accommodate is a globular protein, especially enzyme catalysis. Enzyme catalysis. Fifth one is a examples the myoglobin. Okay. Next one, structure of protein. The proteins are the polymer of an amino acid. First point number one. The proteins are the polymerization of an amino acid. First number one. Second one, it is a three-dimensional structure. It is a three-dimensional structure. Okay. First, primary structure. There are 4 major structures. First one, primary. Second one, secondary. Third one, tertiary. Fourth one, quaternary. First one is a primary structure. So, primary structure of a protein. Primary structure. First one, the primary structure is contained polypeptide chain. Polypeptide chain. So, polypeptide chain. First number one. Second one, so many of the amino acid. Many of the amino acids contains the primary structure become polypeptide chain. Polypeptide chain. So many of the amino acid it contains, that is NH2 and COOH. Already you know peptide bond it contains one acid group is reactive with the amino group. One acid group is reactive with the amino group. Example, glycine. Glycine. So glycine becomes NH2. Glycine. Last one was COOH acid. Ascorbic acid. Ascorbic acid. So ascorbic acid, that is acid. So this is an acid. This is amino. So acid and amino. It's the removal of water. Removal of water. The structure become this is a peptide. More than one amino acid. Already you know the protein is nothing but the polymerization of an amino acid. So more than one amino acid, this is called a polypeptide. Polypeptide. Next one, secondary structure of proteins. Secondary structure of protein. First, the secondary structure of protein, high regular shape. High regular shape. So high regular shape of an secondary structure. Second number, point number 2, it contains COOH, acid group and as well well as a NH2 group. Acid group and as well as a NH2 group. When a secondary structure further classified to, first number 1, alpha helix. Alpha helix. So in this alpha helix, arrangement of on right hand side. The proteins on arrangement of on right hand side. Number 1. Second point number 2, carbonyl oxygen atom, 1 amino acid, that is nth residue. Carbonyl oxygen atom, 1 amino acid, that is nth residue. Suppose amino acids kuda fifth position, that is n plus fourth residues. N plus fourth residues kuda. Each third alpha helix, each third alpha helix thirds 3.6 residues. One more question. Each third alpha helix, the range become 3.6 residue. Next one size 5.4 Armstrong is a long. 5.4 Armstrong is a long structure. Fifth one, it is a cyclic one. Fifth one, it is a cyclic. This secondary structure alpha helix becomes high regular shape. High regular shape. So one of the end would have the amino acid end, another one of the end would have the one of the end would have N, another one is here, that is COOH. This is a alpha helix. Next go to beta strands. Beta strands. Beta strands first number 1, peptide chain rather than to cold conditions. Peptide chain rather than to cold condition, beta strand. Second point number 2, it contains is a hydrogen bond is occur in nature. Beta strands is contained, it is contains a hydrogen bond occur. Third one, so mainly the carbonyl groups are present. So main carbonyl groups are present. Fourth one is the arrangement. The arrangement of one protein contains beta sheet. The structure of the beta strands. Beta strands. Next one, tertiary structure. Tertiary structure. It contains a secondary structure. It contains a two secondary structure of element like alpha helix and beta strands. Alpha helix and beta strands. The tertiary structure of protein contains three-dimensional arrangement. Three-dimensional arrangement. First number 1. Second one, it contains a polypeptide protein. Polypeptide protein. So secondary structure of alpha helix and beta sheets. Beta strands. Alpha helix and beta strands. Three-dimensional arrangement of polypeptide protein is known as tertiary structure. Tertiary structure. Next one, in this tertiary structure become interaction between side chain. Interaction between side chain of one amino acid. Side chain of amino acid. Next one, it contains a disulfide group. Disulfide bridges between cytosine, hydrophobic, hydrogen bond and weak van der Waals forces. Weak van der Waals forces. This is known as a tertiary structure. So tertiary structure is contains alpha helix and beta strands. Alpha helix and beta strand. It is a three-dimensional arrangement. So three-dimensional arrangement of polypeptide bond is known as tertiary structure. Next one is interaction between the side chain of amino acid. Tertiary structure, interaction between side chain of an amino acid. So it contains a disulfide bond. Disulfide bridges between cytosine, hydrogen bond, weak van der Waals forces. Okay. Next one, quaternary structure. So quaternary structure becomes only polypeptide chain. First number 1, it contains only polypeptide chain. Second one is the oxygen transporting of a protein. It's a oxygen transporting of the protein. Second, point number 2. Point number 3, hemoglobin. Hemoglobin contains 4 peptide chain. Hemoglobin contains 4 peptide chain. Fourth one, DNA. So DNA polymerase. DNA polymerase enzyme that makes copy of DNA. Sir, DNA is a polymerized enzyme that makes a copy of DNA. This is known as quaternary structure. Okay, so quaternary structure is contains one polypeptide chain, oxygen transporting protein. Oxygen transporting protein. Hemoglobin contains 4 peptide chain. 4 peptide chain. Fourth one is a DNA polymerized enzyme that make a copy of DNA. Okay. Finally, we are conclude. Is a amino acid is a primary structure. This is a primary. So primary structure. This is an amino acid. This is a primary structure. So secondary structure. This is a alpha helix. Alpha helix. This is a secondary structure of one protein. Third one, polypeptide chain. This is a tertiary structure of one protein. This is a polypeptide. So polypeptide chain, it is a tertiary structure. Next, fourth one is a quaternary. Quaternary structure, that is complex of one protein molecules. Complex of and protein molecule. This is a structure of one protein. There are 4 major structure. First one, primary structure. Second one, secondary structure. Third one, tertiary structure. Fourth one is a quaternary structure. Okay. Thank you.